In this video, we'll be talking about the cell, the fundamental unit of life. What is a cell? Cells are the basic structural and functional units of living organisms. In other words, cells make up living things and carry out activities that keep a living thing alive. Structure of a cell. The detailed structure of a cell has been studied under compound microscopes and electron microscopes. Certain structures can be only seen under an electron microscope. The structure of a cell, as seen under an electron microscope, is called an ultrastructure. An animal cell seen in this image contains nucleus, Golgi body, vesicle, plasma membrane, mitochondria, cytoskeleton, centriole, lysosome, cytoplasm, rough endoplasmic reticulum, also known as RER, smooth endoplasmic reticulum, also known as SER, and nucleus. A plant cell shown in this image contains a nucleus, Golgi body, vesicle, lysosome, plasma membrane, mitochondria, chloroplast, cell wall, vacuole, smooth endoplasmic reticulum, rough endoplasmic reticulum, and a nucleus. A bacterial cell shown in this image contains capsule, cell wall, plasma membrane, cytoplasm, flagellum, food granule, plasmid, ribosomes, nucleoid, and pili. If we study a cell under a microscope, we would come across three features in almost every cell: plasma membrane, nucleus, and cytoplasm. All activities inside the cell and interactions of the cell with its environment are possible due to these features. Now, what is plasma membrane? Plasma membrane is extremely delicate, thin, elastic living, and semi-permeable membrane. It is made up of two layers of lipid molecules in which protein molecules are floating. Thickness varies from 75 to 110. It can be observed under an electron microscope only. Functions: maintains shape and size of the cell, protects internal contents of the cell, regulates entry and exit of substances in and out of the cell, and maintains homeostasis. Cell wall. A cell wall is the non-living and outermost covering of a cell. It can be tough, rigid, and sometimes flexible. It's made up of cellulose, hemicellulose, and pectin. May be thin or thick multilayered structure. Its thickness varies from 50 to 1000, and it provides definite shape, strength, and rigidity, prevents drying up, helps in controlling cell expansions, and protects cell from external pathogens. Nucleus. A nucleus is a dense spherical body located near the center of a cell. Its diameter varies from 10 to 25. It's present in all the cells except for red blood cells and sieve tube cells. It's well developed in plant and animal cells and undeveloped in bacteria and blue-green algae. Most cells are uninucleated, having only one nucleus. Few types of cells have more than one nucleus. The nucleus has a double-layered covering called nuclear membrane. Nuclear membrane has pores of diameter about 80 to 100. Colorless dense sap present inside the nucleus is also known as nucleoplasm. Nucleoplasm contains round-shaped nucleus and network of chromatin fibers. Fibers are composed of DNA and protein histone. These fibers condense to form chromosomes during cell division. These chromosomes contain stretches of DNA called genes. Genes transfer the hereditary information from one generation to the next. Functions: it controls all cell activities like metabolism, protein synthesis, growth, and cell division. Nucleus synthesizes RNA to constitute ribosomes, and it stores hereditary information in genes. Cytoplasm. Cytoplasm is jelly-like material formed by 80% water. It's present between the plasma membrane and the nucleus. It contains a clear liquid portion called cytosol and various particles. These particles are proteins, carbohydrates, nucleic acids, lipids, and inorganic ions. It also contains many organelles with distinct structures and functions. Some of these organelles are visible only under an electron microscope. It's granular and dense in animal cells and thin in plant cells. Endoplasmic reticulum. It is a network of tubular and vesicular structures which are interconnected with one another. Some parts are connected to the nuclear membrane while others are connected to the cell membrane. There are two types. The first type is smooth because it lacks ribosomes, and the second is rough because it is studded with ribosomes. Functions: it gives internal support to the cytoplasm. RER synthesizes secretory proteins and membrane proteins. SER synthesizes lipids for cell membrane. In liver cells, SER detoxify drugs and poison, and in muscle cells, SER store calcium ions. Golgi body. It was discovered by Camillo Golgi. It is formed by stacks of 5 to 8 membranous sacs. Sacs are usually flattened and are called the cisternae. It has two ends: the cis face situated near the endoplasmic reticulum and trans face situated near the cell membrane. Functions: it modifies, sorts, and packs materials synthesized in the cell. It delivers synthesized materials to various targets inside the cell and outside the cell. It produces vacuoles and secretory vesicles. forms plasma membrane and lysosomes. This image shows the Golgi body at work. Lysosomes. Lysosomes are small spherical single membrane sacs. They are found throughout the cytoplasm and are filled with hydrolytic enzymes. They occur in most animal cells and a few types of plant cells. Functions. They help in the digestion of large molecules, protect cells by destroying foreign invaders like bacteria and viruses, and help in the degradation of worn-out organelles. They also perform autolysis in dead cells. Vacuoles. Vacuoles are single membrane sacs filled with liquid or sap. In animal cells, vacuoles are temporary, small in size, and few in number. In plant cells, vacuoles are large and more in number. They may be contractile or non-contractile. Functions. They store various substances, including waste products, maintain osmotic pressure of a cell, and store food particles in amoeba cells. They provide turgidity and rigidity to plant cells. Mitochondria. Mitochondria are small rod-shaped organelles bounded by two membranes, inner and outer. Outer membrane is smooth and encloses the contents of mitochondria. Inner membrane is folded in the form of shelf-like inward projections called cristae. Inner cavity is filled with matrix, which contains many enzymes. They contain their own DNA, which are responsible for many enzymatic functions. They synthesize energy-rich compound, ATP. ATP molecules provide energy for the vital activities of living cells. Plastids. Plastids are double membrane-bound organelles found inside plants and some algae. They are responsible for activities related to making and storing food. They often contain different types of pigments that can change the color of a cell. Chromoplasts are plastids that produce and store pigments. They are responsible for different colors found in leaves, fruits, flowers, and vegetables. Here are a few examples. Leucoplasts are colorless plastids that store foods. They are found in storage organs such as fruit, tubers, and seeds. Chloroplasts. They are double membrane-bound organelles found mainly in plant cells. They are usually spherical or discoidal in shape. They show two distinct regions, granule and stroma. Granule are stacks of thylakoids. Thylakoids contain chlorophyll molecules which are responsible for photosynthesis. Stroma is a colorless dense fluid. Functions. They convert light energy into chemical energy in the form of food and provide a green color to leaves, stems, and vegetables. Centrosome. Centrosome is the membrane-bound organelle present near the nucleus. It consists of two structures called centrioles. Centrioles are hollow cylindrical structures made of microtubules. Centrioles are arranged at right angles to each other. Functions. They form spindle fibers which help in the movement of chromosomes during cell division and help in the formation of cilia and flagella. Cytoskeleton. Cytoskeleton is formed by microtubules and microfilaments. Microtubules are hollow tubules made up of a protein called tubulin. Microfilaments are rod-shaped thin filaments made up of a protein called actin. Functions. It determines the shape of a cell, gives structural strength to the cell, and is responsible for cellular movements. Here is a comparison between a prokaryotic cell and a eukaryotic cell. In prokaryotic cells, a nucleus is undeveloped, only one chromosome is present, membrane-bound organelles are absent, size ranges from 0.5 to 5. An example would be bacteria and blue-green algae. In eukaryotic cell, a nucleus is well developed, more than one chromosome are present, membrane-bound organelles are present, and size ranges from 5 to 100. An example would be all other organisms. Here is a comparison between an animal cell and a plant cell. Animal cells are generally small in size, cell wall is absent, plastids are absent, vacuoles are smaller in size and less in number, and centrioles are present. In plant cells, they are generally large in size, cell wall is present, plastids are present, vacuoles are larger in size and more in number, and centrioles are absent. Thank you for watching.